Hi, today we're going to be talking about the process of DNA replication. DNA is composed of the macromolecule nucleic acid. Nucleic acid is composed of the monomer and nucleotide. A nucleotide monomer is made up of a phosphate group, a 5-carbon sugar, and a nitrogenous base. There are 4 different types of nitrogenous bases that make up DNA: thymine, cytosine, adenine, and guanine. DNA is composed of 2 strands of nucleotides. These strands run anti-parallel, going from a 5' to 3' end to 3' to 5' end. The 5' end ends in a phosphate group, while the 3' end ends in a hydroxyl group. The first step of DNA replication occurs when the enzyme helicase divides the 2 strands by breaking the hydrogen bonds between the 2 bases. This creates the replication fork, which becomes the site of DNA replication. After that, a single-stranded binding protein attaches itself to the bases in order to prevent the bases from rejoining. The first step is when the enzyme primase attaches itself to the strand. There is an enzyme that attaches nucleotides, RNA nucleotides, to the DNA strands. The primase enzyme establishes an RNA primer, which serves as the seat for the DNA polymerase III. DNA polymerase III is unable to attach directly to a DNA strand, thus it needs the RNA primer in order for it to sit onto the primer and attach to the strand. The DNA polymerase III attaches individual nucleotides to the old parent strand. These nucleotides form hydrogen bonds with the existing nucleotides to create the new strand of DNA. Once DNA polymerase adds nucleotides to the strand, it is time to get rid of the RNA primers. The enzyme DNA polymerase I comes in and it replaces the RNA primers with the corresponding DNA nucleotides, thus completing the strand. Replication of the lagging strand first begins when the enzyme primase attaches itself to the strand and attaches RNA primers to the current DNA strand. RNA primase creates fragments of RNA primer. Due to the fragmented nature of the primer, when the DNA polymerase III attaches itself to the RNA primer in order to create the DNA strands, fragmented DNA strands are created. These fragmented strands are called Okazaki fragments. When the DNA polymerase III attaches itself to the next segment of RNA primers, it creates a new fragment of DNA nucleotides. Once the DNA polymerase III is finished adding nucleotides, DNA polymerase I comes in and replaces the current RNA nucleotides with their corresponding DNA nucleotides. The current DNA strand is divided into its Okazaki fragments, fragment 1 and fragment 2. Next, the enzyme ligase comes in and creates a phosphodiester bond between the nucleotides of both fragments. This creates one long strand of DNA by joining both fragments. Thus, ligase joins the 2 fragments, creating the one long strand of the new DNA, which marks the end of the DNA replication process. In some of the DNA replication process, the enzyme helicase divides the DNA strand into 2 strands. This creates the replication fork and the site for DNA replication. The enzyme primase attaches itself to the DNA chain and creates an RNA primer. This primer serves as the base for DNA polymerase III. Since DNA polymerase III is unable to attach itself directly onto the DNA strand, it sits on the RNA primer and begins attaching nucleotides to the current strand. Once this process has been completed, DNA polymerase I comes in and replaces the RNA primers with new DNA nucleotides. The DNA replication process occurs on 2 strands, the leading strand and the lagging strand. Since the DNA replication has to follow the model from a 5' end to a 3' end, the leading strand is built leading up to the replication fork following 5' to 3', whereas the lagging strand is built away from the replication fork following the 5' to 3' end model. The leading strand is created continuously, whereas the lagging strand is created in fragments. These fragments are known as Okazaki fragments. Thus, the enzyme ligase is needed to join the fragments by creating phosphodiester bonds through the nucleotides of each fragment. This sums up the process of DNA replication. Thank you for watching.